Today we're going to be talking about protozoa, the intestinal amoeba. Some general information about protozoa. They are single-celled animals. Most species are free-living. Now some protozoa have structures for propulsion and other types of movement. For example, pseudopods, flagella, and cilia. Many multiply by binary fission. Some use sexual and asexual reproduction. Most have two stages, while some have a single stage. And the stages are known as this: the trophozoite stage. This is the motile, reproducing, feeding, and infective stage. And it's typically in an irregular type shape. Then there's the cyst stage, which is the non-motile, non-feeding, infective stage. It's dormant and is typically round. This next diagram just gives you a demonstration of the trophozoite, how it encysts, to become a cyst, it's passed in the feces, and it's excyst. And that's what you typically see with the intestinal amoeba that we're talking about today. Now the first one I'd like to talk about is Entamoeba histolytica / dispar. And this is two separate species of parasite that are very difficult to differentiate. Now all of these amoeba we're going to be talking about today have a cosmopolitan distribution. They're all contracted via the ingestion of contaminated food and water. But only one is a true pathogen, and that's Entamoeba histolytica. And you can identify that microscopically by seeing ingested red blood cells within a trophozoite. Entamoeba histolytica is associated with both intestinal and extraintestinal infections. Now, Entamoeba dispar, which is a non-pathogen, is morphologically identical to E. histolytica. The pathology of E. histolytica is known as amoebiasis, which is amoebic dysentery, and you may see flask-shaped ulcers of the colon. If the amoeba leave the intestinal tract, we call that extraintestinal amoebiasis. And this is very serious infection, and it can go to the brain, the liver, the skin, and the lungs. This is the life cycle for Entamoeba histolytica, and other than the fact it has parts for extraintestinal disease, this life cycle is the same for all the amoeba we'll be talking about today. Let's talk about the internal structures for Entamoeba histolytica. The nucleus. You're going to have a small central karyosome and a fine, even peripheral chromatin. Chromatoid bodies are going to be with rounded ends, and it's frequently called cigar-shaped. Cysts will have between 1 and 4 nuclei, a mature cyst having 4. The cysts usually measure about 12 to 15 microns. The trophozoite is only going to have one nucleus, usually measuring about 15 to 20 microns, but it can have a much wider range. If you do see the ingestion of red blood cells, this can be used to differentiate E. histolytica from E. dispar. The trophozoites of E. histolytica have a progressive motility. And in these pictures you can see the E. histolytica, E. dispar cyst, arrows pointing at the nuclei and the chromatoid bars. But based on what we see in this picture, you cannot differentiate the two. And here we have the E. histolytica, E. dispar trophs. Again, a single nucleus. Again, you cannot differentiate between the two species. However, when you get to these photographs, you can, because you can see the ingested RBCs within these trophozoites. So this is Entamoeba histolytica. And something that you may see with Entamoeba histolytica when you're examining a stool prep is something called a charcoal-laden crystal. And you can see that on the right. Those are those elongated diamond-shaped crystals that could be found in the stool. And these are the breakdown products of eosinophils. They are slender and pointed at both ends, normally colorless, but they will stain red when you're dealing with a trichrome stain. Let's talk about the commensals real quick. Just some general information. There's a handful of them. Entamoeba coli, Entamoeba hartmanni, Entamoeba pollecki, Endolimax nana, and Iotamoeba bushlii. They're all non-pathogenic, and they all reside in the large intestine of the human host. If they're non-pathogenic, why do we care? Because for a person to have this, they had to ingest tainted water or food. So though these are non-pathogenic, the potential for having a pathogen somewhere in the stool still exists. So that's why we still report it out. Now, both the cyst and the trophozoites of these species are passed in stool and considered diagnostic. Entamoeba coli, the cyst stage. Now, this is the largest intestinal amoeba. Usually it's spherical, but may be elongated, measuring 10 to 35 microns. Mature cysts typically have 8 nuclei, but they may be hypernucleated and they can have up to 16 or more. Entamoeba coli is the only species in the genus that you're going to see more than 4 nuclei in the cyst stage. Looking at the more internal structures of the nucleus, you typically have a large, smudgy, eccentric karyosome with a coarse, irregular peripheral chromatin. If chromatoid bodies are seen, they are going to appear splinter-like with pointed ends. Now, the chromatoid bodies are seen less frequently than in E. histolytica. Now, in these photographs, we can see cyst stage of Entamoeba coli, and you can see that there's more than 4 nuclei in each one of these photographs. Now, the trophozoite of E. coli is very large, usually measuring in a large range of about 15 to 50 microns. They will have a single nucleus with a large eccentric karyosome, a coarse, irregular peripheral chromatin, and the cytoplasm is usually coarsely granular and vacuolated, often described as a dirty cytoplasm. Motility is different from Entamoeba histolytica in the fact that the motility of Entamoeba